All right. Thank you, everybody, for having me here today. For those of you who don't know, I'm Maggie Casal. I work at the University of Pennsylvania. I got my vet degree in Switzerland a long time ago. I've been doing this for a really long time. I came to the United States for a 2-year residency and never left, so in medical genetics. Clinically, I see the only patients I see anymore are pediatric patients, pediatric dogs and cats with bizarre genetic diseases or other diseases. Anyway, so I'm going to talk about, I hope you can see this. I don't have any pictures of Sealyhams, but I do have a picture of my late dog that I really love. So how do we develop these kind of genetic tests? First, we have to rely on, and I think Dr. June said that perfectly, we have to rely on clinical signs. We have to get the videos. We have to get as much information as we can, because we were very unfortunate that we didn't get really good information from the histologic examination, so under the microscope examination of the tissues. So it's a very complicated disease in that sense. So pathology, like I said, we did have some pathology results come back, but it says it probably means as much to you as it means to me, non-inflammatory myopathy. It means there's a muscle disorder where there's no inflammation. Okay, well, that's not helpful. Muscle fiber degeneration and regeneration. So the muscle fibers broke down and they tried to get it to come back. Not really helpful either. And so ultimately we were given a diagnosis of some congenital polymyopathy, which means congenital means born with. So a disease where you're born with a polymyopathy means just a whole bunch of muscles are affected. Again, not really super helpful for a geneticist that's trying to find a gene that causes this, because this could be pretty much anything. And then we were given the other diagnosis, congenital myasthenic syndrome. And in fact, Dr. Shelton's own words were some form of congenital myasthenic syndrome. So we're trying to work on trying to figure out what's going on. And I think what's going to happen is, like we were saying before, this is I don't think this is a regular autosomal recessive disease, meaning it's going to be a little more complicated. But what I'm hoping is that it's going to be a dominant disease, and I'll talk about that a little bit. With incomplete penetration means that some dogs are actually affected, but you don't see it. And so that would explain why you have these varying degrees of affectedness. Okay, so let's talk a little bit. We already talked about autosomal recessive, but I wanted, and you already know this now. So you have a big A and a little a, meaning big A is the dominant form of the gene, little a is the recessive form of the gene. So when you have little a, big A in one parent, little a, big A in the other parent, you get exactly that Punnett square that you already saw. So a quarter of your offspring will be affected. This doesn't seem to be the case. Like a lot of times it looks like you have multiples affected in one litter, which would actually speak for a dominant disease. If you're looking at autosomal dominant, so my pedigrees, I convert all of my pedigrees into this, because this is, and I convert all of the dogs' names into numbers, because I can't remember all the fancy names, but I can remember numbers. So how does a dominant disease work? Squares are males here, circles are females. Filled in symbols are those that are affected. And this is not your breed. This is another example. So this is not yours. But the filled in symbols are the affecteds, and the open symbols are those that do not show clinical signs. And so what you can see here is you have a female. Sorry? Okay. All right. So you have a filled in female up top there that is affected. And then you will see she's bred to the male all the way over to your left. And then you can see in this first litter, there's 3 males that are normal, 2 females that are affected, and another female that's normal. But what's most important to note is that for every litter where there's affecteds in it, 1 parent is affected. That's very classical for a regular dominant disease. Where do we have dominant diseases? I don't know if I have this on the next. Oh, it's right on there. What breed is that? Chinese crested. That is an autosomal dominant disease. Okay, I call it a disease because it is actually one. But yes, it's a breed. So it's a trait that's like, in this case, your dominant affected breed can breed, which is fine. But what do breeders of Chinese cresteds never do? You got it. So you never breed to two affecteds together. You always breed one to a powder puff, which is the haired version of the Chinese crested. Why do you do that? Because if you breed two dogs, Chinese cresteds together, a quarter of the offspring will have a double dose of that dominant disease, dominant trait. Sorry, dominant trait. And then the big D, that's a double dose. Those die in utero. Those never make it to birth. So here's a trait that's kind of desired, a dominant trait, but it is actually this is why I call it a disease, because if you breed two together and you have a double dose, they die before they even make it to this world. So that's something that you can see. And there are other disorders also that are traits that are desired, but if you double up on them, and those are some of the dominant disease. Sometimes you get dominant diseases that are truly diseases. So, for example, everybody knows Abraham Lincoln, right? So Abraham Lincoln, what did he have? Marfan syndrome. That is a dominant disease. So I know a family that has Marfan's where sometimes when the father was pretty affected and his daughter was very, very minorly affected, and her sons were severely affected. So there's a dominant disease where you can see it kind of varies as it goes through. So that's what's called incompletely penetrant, or like there's other words for it, but in any case, it's like a dominant disease that doesn't always show itself like full on. So that's something. But how do I go about working up these things? And this is why you'll see these things can't be done from like in 24 hours. They're pretty complicated. But I'm going to make a geneticist out of all of you. Okay, so learning objectives here are principles of DNA for those that are less familiar with it. What is a marker test? That's important for everybody to know, because you have some tests out there that are marker tests and some that are mutation tests. And how do we find a mutation? So that's what we're going to talk about here. So what is DNA? DNA consists of building blocks. And I, like Dr. June, I don't work with complicated stuff like that. I like to convert everything into analogies or easy ways to look at it. And you'll see that in a second. So building blocks of DNA there are four what are called base pairs. And for short, they are A, C, G, and T. These are the building blocks. So four different ones. Imagine these as four different colors. So we're going to use glass beads to substitute the little Indian beads that we all played with. We're going to use those to substitute for those nucleotides that we just talked about, the A, C, G, and T. How long do you think a dog's DNA would be if the A, C, G, and T's were each a different color of glass bead? How long do you think the DNA is? 100 yards? Yes, Philadelphia to San Francisco. That is how long your chain of beads would be. That's a single genome of a dog. And that's not even the double stranded. That's just one strand of DNA. That's how long it would be. Now let's look at this a little closer. So that's to put in perspective. There's your DNA strand from a dog. It's similar for people. It's a little bit longer for people. But so how if you had to calculate it out using the glass bead analogy again, we have about 20,000 genes that make up a dog. And each of these genes would be about 20 to 30 feet long. However, how far apart are they? They're about 600 to 800 feet apart, again, using the glass beads, so you can sort of imagine. This is a Philadelphia city block. Unlike New York City blocks, it takes 8 Philadelphia city blocks for a mile, whereas in New York it's 10. So you can see what I'm trying to show is, and I do this with the students too. So the little arrows here, the yellow arrows, they designate the gene. The genes are sitting sort of at the stop signs at the end of each block. And then you have all the parked cars in between. These are what are called markers. And markers are just little changes in the DNA that act like they're genes, but they're not. They don't code for anything, but they behave inheritance-wise like a gene. And so you'll see that in the next couple of illustrations. So genes code for a protein. They code for something. They code for eye color, hair length, hair curliness, the furnishings, all of those kind of things. But markers don't really code for anything. But like I said, they're little. So if you imagine these are a pair of chromosomes in the dog, and you have G for green, O for orange, B for blue, P for purple. So these are like random markers that are all the way throughout the gene. But then if you look at them a little closer, you will see that they actually have little variations. So here we have so those are genes over there. Here we have the blue gene and the purple gene, and then you have all these markers in between. And the markers, like I said, they'll be inherited just like a gene would be. So if one marker has 16 copies and the other marker has only 8 copies of a CA repeat, for example, and you inherit those, you'll see that on the next slide, how they match up. Okay, so did I miss this? No. To make things really complicated, and this is why markers fail. Markers, there's such a phenomenon called crossing over. And what can sometimes happen, if you imagine these are chromosomes, if you imagine that, what happens if you put the wrong tip on the wrong marker? Okay, so this is basically what's crossed over. So this can happen with a chromosome too. So you can have a part of a chromosome will go to its mate chromosome, and the tops will get flipped. What that means is the markers, and you'll see that in the next couple slides, the markers that we're looking at now all of a sudden are no longer associated with the gene that you had it before. And so you'll see, so you'll see what happens. So these are again your pairs of chromosomes, right? There you have the dam up there, and she has a gene A up there, big A coding for something. So this is in an autosomal recessive case. And then you have the little a coding for the recessive form of that gene. Marker 1 traveled with big A, marker 2 traveled with little a. And here in the sire, what happened was all of a sudden there was a crossing over event that happened at some point, and now marker 2 is associated with big A. So it's no longer an accurate marker that we use. Why do we even use markers? We use markers when we don't know where the defective gene is. So what we do is, and you'll see that in the next couple slides, we go through and we try to find where markers, we know where all the markers sit. And so what we usually do is we go and look at which markers are present in the affected animals and which markers are not. And so we say, like, okay, so we found marker 764. We found, oh, that always travels with the affected animal. And so we see that, and then we say, like, that means the gene has to be nearby. Because it's easy for us to look for markers. That's easy. But to look at the single genes, you'll see we go about it a slightly different way. But when we find that marker that's always there with the affected animal, we then know that gene has to be nearby, and that's where we have to go look. And a lot of times, what companies will do, or sometimes we even do that, is before we have found the mutation, but we want to help, we want to get the test out there as quickly as possible, we will sometimes offer a marker test. And you know now that the marker test can actually be wrong because of that crossing over. So if we look at the different markers, there's another way to look at it. So if you have blue here, that's the gene with its important parts and its less important parts in between. So you have your gene there, and you have a variety of markers, marker 1, 2, 3, 4, and 5. So which marker, if I had to pick one of these, which is going to be the most accurate marker? 3, right. 3 will be the most accurate because it's sitting in the middle of the gene. So that marker's there, and so that one's probably never going to be wrong. But marker 5 or marker 1, they're so far away from the gene that it can happen that you'll get the crossing over that will happen right here and break off that piece of chromosome and split it over to the other side. So that's how these things can happen. So this is now you understand why when you get a genetic test back, you have to look if it says, is it a marker or a mutation test? A mutation test is going to be accurate, with one exception, and I'll show you that, and that's usually user error. But otherwise, the marker test can actually be wrong. So here's an example. This is a disease I studied a while back. This was ichthyosis in the American bulldog. We found that, again, the squares are males, the circles are females. The sort of half shaded ones are carriers. You can see that we designate them, and you'll find that on some of the genetic tests you get back, you'll get a 1-2 result. So 1 meaning the one allele, 2 meaning the other form of the same gene. And so you can see that 2 2s are affected. And so this follows a pattern. You have 2 carriers, you produce affecteds, autosomal recessive. But what happened here in this case, and this was a marker at the time, we now know the mutation, which is what we test for. But we got this result back. That dog over there, the 2-2 female, all the way over there, that was an Australian dog. So we thought, it can't be 2-2. The dog has no clinical signs. In fact, we were so sure that something's not right here. They sent us a biopsy from the dog all the way from Australia to prove that she was not affected. So you can see, so that was a marker that the crossing over happened. So she was truly a carrier and not affected. But because it was just a marker test, it now made it, because of the crossing over, it made it into a I don't want to say wrong diagnosis, but an incorrect diagnosis because of the crossing over. So again, marker tests can be wrong, depending on how far away the marker is from the actual gene. Mutation-based tests, super unequivocal, always correct, except what happened here. This was a female carrier. She knew the female was a carrier, the breeder, and she did everything correctly. She went and looked for an unaffected, a non-carrier male. So it had to be normal. So big A, just normal copies of the gene, because that way she could preserve. This was in Newfies, in Newfoundland. She could preserve the really good standard that she had of the breed. This was like a super awesome female. She wanted to keep it, but she didn't want to produce affected puppies, which is fine. And so she specifically sought out a male that was not a carrier. And all of a sudden, an affected was born in the litter. How did that happen? That is not the father. So what happened was that wherever she had her female bred, they did not breed to the dog that she chose. Yes. And so I'm saying, like, the test is always going to be correct. If you get a result like that, then it's user error in that sense. So yes, that was a very unfortunate situation. So mutation-based tests, like I said, they're generally very accurate. You just got to make sure, like if you're testing puppies and you want to do cheek swabs, you have to take them away from each other and from the mom for a couple of hours, because otherwise, if you get the mom's milk in their mouth and you do cheek swabs, you can actually mix up the DNA from the cells from the mom and the puppy, and then you get incorrect results. How much time did you say you want them off, Mom? 2 hours at least, yeah. And then give them some water in between just to get some rid of that stuff. And then they can't be playing with each other either, because they lick each other, they bite each other. Yeah. So to show you how, to tell you something else about mutation tests, oftentimes you will find these tests like Embark or Wisdom Panel. The Embark test is still one of the best ones out there, but you will find Embark and Wisdom Panels and Paw Print Genetics and all those. They'll offer all these tests. We test for 475 diseases. Do they exist in your breed? No. And this is why I want to show you, so just so you know when you're reading the results, like read them with a grain of salt. This is a disease called mucopolysaccharidosis type 7, which you don't need to know, but it's a pretty severe disease in people as well. This is most of my research, actually, developing treatments for children with these diseases. But anyway, so this is the gene that codes for the protein that's important, for the enzyme that's important in this disease, that you have to have, otherwise you have that disease. So this is a sequence out of this gene, and there's a German Shepherd. This was the original mutation that was found. So here, instead of having a G, there was a C, and that's what caused the disease. So here you have a mutation for the German Shepherd for this disease. A poodle showed up one day with clinical signs that were almost exactly the same as the German Shepherd. And so they went and did one of the tests, one of the commercially available tests, and they said it isn't MPS 7. And they called us, and it's like, yes, it is MPS 7 because I tested the enzymes and I did all the testing to prove that it is MPS 7. Oh, but it didn't have the mutation. Yeah, because your poodle has a different mutation. So all of these tests are very, very breed specific. So you can have in the same gene, you can have PRA, so progressive retinal atrophy, for example. There's one gene where we have 20 different mutations in 20 different breeds. So same gene, but different mutations that cause the disease. So when you're doing these kind of tests, make sure it's the test that's right for your breed.
There have to be test on the eyes, and it was qua B. And they test all the dogs, but we test from the kennel club, so from the club, and they test all the dogs, and a lot of dogs were carrier, of course, and affected. And the people start to sterilize all these dogs, and then 2 years later, it was probably one, because this other test was not specially for Schnauzer.
Yeah.
So they sterilize a lot of all the best dogs for nothing.
Yeah.
And we say be careful because this dog is carrier, you can use it, and after that these carriers were all free.
Yeah.
So it was
Yeah.
right.
There's similar examples from the Portuguese water dogs. There was a disease, a neurologic disease that existed in the Portuguese water dogs in like the 1960s. And the only way to diagnose it was by a specific protein assay or enzyme assay. And that was done. And then the problem with the enzyme assays is a normal dog has 100% enzyme, a carrier dog has 50% enzyme, and an affected has 0. Well, that's in theory. What really happens is that the affected dogs would have between 0 and 20% enzyme activity. Carrier dogs would have somewhere between like 25% to sometimes almost 80% of enzyme activity. And the normals would have somewhere between 65 and 100% activity. And so we didn't know that until, it wasn't my lab or anything that did that, but it wasn't really understood until later, till they had genetic testing. But what they did was they did the same thing. The Portuguese water dog breeder said, like, anything that remotely looks like a carrier is going to be kicked out. What happened? They were left with 6 founding dogs. And what happened? They have now heart disease, they have eye disease, they have microphthalmia, which I just solved. So they have a number of diseases that they didn't have before. And it's not that they created those diseases, but because they were left with such a small stock, all these diseases come out eventually. So one has to be careful. Anyway, so how do we go about finding the gene and its mutation? There's 2 principal ways that we can do this. One is by what's called a genome-wide association study, and I told you I was going to make geneticists out of you. You'll see how that works. And then the other is whole genome sequencing. Basically, whole genome sequencing, you'll see that I think I have an example of that too. So how do we do genome-wide association studies? We use markers. Like I said, there's way more markers out there than there are genes. And we know most of the markers. They have all fancy names with letters and numbers. We know where they are. So what we do is all the little markers, this is a glass slide, like the size of a microscope slide, has little tiny, over 100,000 little pieces of DNA that are equivalent to the markers. And so what happens is that when you chop up the dog's DNA and throw it on there, then the markers will stick. So if you have one version of the marker, it might stick to its counterpart. And if you have the other version, it might stick to the other counterpart. They're all labeled with a color, and it's going to be a red or a green. And we know which ones are labeled which way. And then this then gets done by, these are machines that do this. This isn't done by hand anymore. But these are machines that do this. And then you get like this huge graph of information. Mind you, you're getting 100,000 pieces of information there. And you do this for 100 dogs, you've got a lot of data to work with. So this is a lot. So what we're looking for is, I'm going to explain how you do this. Anybody can do a genome-wide association study. you like these donuts. They're called Frank's Donuts, right? And so, but you really want to go visit the factory, but you don't know where the factory is located because it doesn't say on the package, but you really want to go look. So you gather up all your friends and you put them on all the highway exits throughout the contiguous United States, right? Give them a clipboard, piece of paper, and a pencil. Every time a Frank's Donut truck goes by, make a little mark. You do this for a whole month. Have everybody sit there for a whole month writing down how often a Frank's Donut truck goes by. And so now what you get is the person in San Diego, so like on average 0.1% of the time a truck went by. Miami, Florida, 0.1, I hope that's where Miami is, I'm not sure. 0.1% of the time that a truck went by. Up Maine on the Canadian border, 0.1% of the time a truck went by. But now look, you have 0.4% of the time, Louisiana maybe, and then you go up towards, as you're going towards Wisconsin, you can see it increases in number. So many more times the truck goes by in a day than it did like down there, right? And so where on the map, where's Frank's Donuts located? Wisconsin, right? Yeah. So in yeah, Milwaukee maybe. Yep. So yeah, so that's essentially how you do a GWAS or a genome-wide association study. You're basically looking which marker shows up the most in the diseased animals or in the animals that are affected with a particular disorder. So now the computer did not do what I wanted it to do. Come. There we go. Okay, so what we're looking for is, these are some other studies that we did for something else. We call this a Manhattan plot. And so you can see all the way to your left, you can see the graph where you have that one peak that stands up. Each dot represents a dog and a marker. And so you can see, and on this axis, on the x-axis, is all the chromosomes are lined up. So you can see there's this huge peak over there. That means there are all of the affected dogs. They all are, that marker's always there. All the other markers are there, maybe, maybe not, but in that area, so you know that's like basically Wisconsin, right? So that's where you have the diseased dogs or the affected dogs are associated with a marker in that area. So what that means is I need to go look at that area more closely, and this is that area blown up, and so I can see here there's an area here where it is. And then I go to the computer and then I go look at what genes, these are all different genes that are in that area. And so I can do one of two things. I can go look at every single gene and figure out, does this make sense or not? Or I can do other studies to more define this. But this is essentially the way I used to do things, is go through and look at every single gene and see if it makes sense for the disease I'm looking at. Sometimes, like in this case, nothing made sense. And so what happened in this case is we actually then did a whole genome sequencing, and we actually found a new gene that has never been described before in causing that disease that we were looking at in Portuguese water dogs. So that was something really new. So we actually discovered a whole new disease-making process, which was rather interesting. So whole genome sequencing, remember all those beads? So they went from Philadelphia to San Francisco. Okay, so there's two ways of doing whole genome sequencing. One is called a low pass and one is called a high pass. And so you have 21 million feet of beads to look at. So what you're going to do is you're going to get 26,000 of your friends, lucky you, and you're going to have them go read stretches of DNA, maybe about 800 feet worth of beads. They're going to tell you every single color in order, right? And you can imagine, you do this 800 beads and you're going to have to write down what colors, you're going to make a mistake. Everybody makes mistakes. So low pass sequencing being that you have your friends read 800 feet of beads 4 times. So now you're cutting down a little bit on the mistakes made. If, bead number 3,600 or so is counted as a green 3 times and then once as a red, it's probably going to be green. But if you do this 36 times, you're going to be way more accurate. So does that so far make sense? And if I ever use words that you don't know, like throw a shoe at me or something, just raise your hand. So, but that's how you do a low pass versus a high pass. So that's what we actually started out. We started with a low pass sequencing to see if we can get a ballpark area. Um, so, so we were not so lucky. This is for that's not should be myasthenia syndrome, not gravis. Um, but what you can see is there's a couple of highlights here that you can see a couple of dots that go up. What we really want to see something is over the blue line. That would be more accurate. But I think like looking at more and more information that we have, um, Leo, the guy I do all my genetics with, he said not all hope is lost. He analyzed it a couple of different ways. You have a small breed, that's to our advantage, because that means there's large stretches of DNA that are going to be very much the same between dogs, and so that helps us narrow down things because we can say like, okay, this is the same in all Sealyhams. We don't have to look at that, or it's less likely going to be here. So this doesn't look as nice as what we saw in the Portuguese water dogs yet. But what we did do, we sent out one affected dog to have him whole genome sequenced 36 times. And so that I think will help us a lot. And then compare that data to what we already have. And in the worst of all cases, we're going to have to send out another 12 dogs. 12 meaning 12, a couple affected, their parents, maybe some siblings. But we have that, and I think we have almost all of the DNA for that if we need it. But that's sort of where we're at. We're supposed to. I was kind of hoping I would get the sequencing back by the time I came here because we sent it out about 3 weeks ago. So I know that within like the next week we should get those results. Still, it has to be analyzed. Remember, you're looking at millions and millions of data points, so it's a lot of computing power and a lot of Leo, my geneticist, being like really good about finding things. Um, so that's sort of where we're at right now, just to give you sort of an overview. But also, I wanted you to understand how kind of complicated it is to do these things, to figure these things out. Although now you know how to do them. So yeah. So what the best area I could find so far is this area, which is on chromosome 1 actually. So I'm still looking through all of these genes while I'm waiting. I can't sit idly by. So while I'm waiting, I'm having fun and looking at all those genes to see if any one of those could be a possible explanation. Because if it is, then that's something that we can very targeted look at very quickly. So, and I think that's all I had. Yeah, go ahead.
So you're waiting on genome sequencing, is that what you said?
Mhm.
And you thought maybe within 3 weeks you might get it. And then what's next
So
after you have those results that you kind of hope to come by now?
yeah, so I think we should be getting them in less than 3 weeks, but it has to be analyzed still. So once we have that, and once we're fairly certain we have found what we think causes the disorder, then at that point we develop a test, and that usually goes very fast. That's like I'll go to the computer and say like I want this is my mutation. I want this in this test. I put it in a program, and within like 10 minutes I'll have a test developed. I order it. It usually gets here within a week, and then we can start testing. But I will say, like I've been hopeful before to send out something that cost me $1,000 and get something back that says like, no, you'll have to test more dogs. So it can go either way. But I do honestly think that within 6 months or so, like I'm hoping faster, but within 6 months, I'm really hoping that we do have a a, a test out there. That would be nice.
I just want to say that's amazing considering I think you contacted her in December. I think that's amazing.
Yeah.
Yeah. Yeah.
And I want to ask you,
Mhm.
because we sent out my whole litter and June's litter, would we know on those animals first?
Oh yeah, absolutely. Yeah, absolutely. The DNA that we already have, that obviously will get. Yeah, yeah, yeah. You'll definitely be the first to know. You'll be the first to know. Yes.
So if we have a test,
Yeah.
is it going to be something that breeders could use definitively? Because we work
Correct. No, we want a definitive test. We have to. No, we have to. We don't. I don't. Because of those marker tests, we've had issues before, and so I don't like releasing marker tests anymore. Like it has to be a yes or no answer.
And is that what the IVDD RISA test is out of UC Davis? Is that a marker test?
I think that's a marker test, if I'm not mistaken. It's the
I'm talking
same.
about risk.
Yeah,
It's a risk test.
yeah.
It doesn't tell us, oh, your dog's gonna have a bad spine.
Well, it's similar like the DM test. The degenerative myelopathy is also very similar to that as well. Yeah.
Is there ever a point where you need to check this to make sure that your test works? Maybe a bit more DNA.
Yeah, this is the tricky part. So this is the tricky part because I think that it may be a dominant disease that's not always fully penetrant. Then the unaffected, you never know if they're truly unaffected. So that makes it a little bit tricky. And so that's why I'm hoping that we figure this out.
So
Yeah.
asymptomatic effect.
Exactly. Yeah, that's another way of saying it. Yeah. Go ahead.
First, just want to thank you for everything you've done and everything you'll continue to
Oh,
do.
thank you. Thank you.
Second, I apologize, but it's a difficult question for you. So, understanding we still have some time before maybe we have a test, right?
Hmm.
What do you think is the most responsible course of action for people who have breeding programs currently until we have some more answers?
That's a really good question. So with autosomal recessive diseases, I always say you even before you've had once you've produced an affected, you know who the parents are carriers. Like that's in a way easy. And then you say like, okay, if you have my standard answer in those cases is if you have like the best dog conforms to the best standards is like really great at what working whatever it is, and then you kind of don't want to throw all those good genes away, and so I pick the mate super carefully, make sure the other one comes out of a line where they've never seen that particular disorder before, and then you can hopefully before there's a test assume that that one's truly normal and not a carrier. With a dominant disease, that makes it really, really tricky if it's a dominant disease that you don't see, that you can sometimes not see. I would say if like I'm really hoping that within the next 6 months we have something for you, then I would say like hold off with those that you know produced it, but don't just spay them just yet or neuter them. For males, it's really super easy. Freeze a bunch of semen, freeze away a bunch of semen, and then if it turns out that, yes, this one was a producer, then you can say like, well, we froze it, chuck it. We don't want that to happen again. But then if it turns out that that male that was frozen is like a non-affected, clinically super normal, then you can say, now we can use it. Kind of the Cavalier King Charles Spaniel people did that for a while, where with the Cavalier King Charles Spaniel, what percentage has heart disease?
High.
90%. What percentage of the Cavalier King Charles Spaniels have syringomyelia, Chiari syndrome? 90%. So what do? The best thing you can do in those cases is they take the males, they won't even breed them until there's the responsible people. Won't even breed them till they're still heart clear at 6 years of age. And up to that point, they may freeze some semen away or do something. So in that case, yeah, but in this is very, very difficult question to answer, honestly, for this particular this disease. Yeah.
I just had a question for you before you went up there. How many of you are all American breeders, except there's somebody here from Hamburg, Germany, and there's somebody from Frankfurt? I said, you've been looking at mostly North American dogs. And I said, well, has there been an incident of this reported in dogs from Europe, Great Britain? I mean, Sealyhams are elsewhere. You said the people in Europe are not as good at reporting these types of things. How many of you in the room you've seen them from abroad?
We know that it's there overseas.
Okay.
So
Been told that, okay.
I didn't want to say it as brutally as that, but so I personally lived in Switzerland for, I don't know, 20 years. And I will know that some breeders are really good about being forthcoming for some things. and I know that, but also I know from the Cairn Terrier world, which I've been working with for the last 25 years, is some diseases are not reported, and some dogs were sold into this country full well knowing that a kidney was missing. And yeah, and so then I think I don't know. I will qualify this by saying I don't know if those people didn't test or didn't do ultrasounds or sold the dog not knowing, but I don't know, and I.
They said the breed internationally worldwide has a very small gene pool.
Yes, that's for sure.
Obviously, there are dogs from other continents that are bred to those here in the United States. So that's part of the discussion.
I can't say anything about Sealyhams in Europe because I just don't know. I can only speak for the Cairn Terriers because there I actually know.
We have different bloodlines, but like I am in Germany, I don't mostly nothing about the dogs from UK because except you. Same line. But from the other dogs in the UK, we know very few, very few. And especially in UK, there are also crossbreeding with the Sealyhams, with Jack Russell. But this is the backyard breeders.
You get quite a few people breeding that aren't involved in pups and stuff, and you don't really know what's going on.
Yeah,
But we do know that there is a dog that has produced effective puppies, long gone dog, but from a kennel that was produced a lot of puppies that went around the world. In the UK, yeah, towards it.
Yeah.
We can't get away from it because we're beautiful dogs.
Samples, but let's have one more question and then we'll wrap up. The samples that you work on, have they only been affected puppies or have you
No.
been working on relatives of affected?
Yes. Yes.
So just parents?
Yes, so it's really important for our kinds of studies, and especially in this case, that we get the affected, their litter mates, and the parents, and the possible grandparents.
Okay.
So that's
So
been really
all
helpful.
of them on the 4 litters have been tested? No.
No, and that's why we started because so I do things backwards a little bit sometimes. Like I usually ask for money up front before doing these kind of things, and I just wanted to get after I saw Roberts, I just wanted to get this done as quickly as possible. So I put out my own money to start this project, and so I've been paying for this, and so I started what I could afford, and which is like let's start with 12 dogs doing a low passes sequencing. So that was affordable, and then we sent out one for whole genome sequencing, which costs, as it says here, for a high pass sequencing, that's about $1,000. So there, if we don't get the answers we need, we're going to send out the next 12.
We've run them in increments of 12.
Okay.
Yeah. So actually, in her possession, she has, I think, 10 samples, 11 samples, and there's still one at my house.
Yeah.
So we can run another 12 very quickly.
Yeah.
Would it help to have dogs that you don't believe have been
No.
affected at all? No, okay.
Not right now. I think when it comes to like once we feel that like we've nailed it, I think what we're going to do is we're going to have a call for specific dogs that we'd like to test just to prove a point. And I know that the guy I work with used to be in Bern, Switzerland. So sorry, Switzerland keeps coming up, but it just so happened it's very coincidental. But he trained under a guy I used to work with when I worked on skin diseases a long time ago, and he has access to the whole genome sequences of a whole bunch of other Sealyhams that are in his database. And so,
like in Germany, before our dogs go into the breed, we have all DNA from the dogs, and all this DNA are in the club.
Oh, good.
So they have it in labo clean.
Yeah.
Like
I know. Yeah, I
have
do.
it for 10 years. So
Yeah, yeah.
It's safe there.
Yeah.
So if there is something, all dogs who are in the breed in Germany are listed there.
Yeah, yeah.
Because we need DNA in Germany, a terrier before it go. It doesn't matter which terrier.
Yeah.
Before it go into the breed, it has to have the DNA
Right.
in this space.
I think I saw 1 or 2 more hands. Leslie?
Yeah, we had previously a project where University of Missouri,
Mhm.
all that DNA is in the library for researchers to use. Is that something that we will look at, or will you?
If needed, we will. Yeah.
Jack,
How big a sample do you need, doctor, in order to?
Oh, for a blood sample. Usually we ask. Today we can get a lot of DNA out of 2 mLs of a purple top tube, so that's not even a teaspoon's worth of blood. So quite affordable for a Sealyham.
so I want to ask a question
Yeah.
because I just alluded to fundraising. I have to put my president hat back on now. You said you've spent out of your own pocket. So at the present time, approximately how much have we spent on your research? And then we can also tell you what we've spent as part of gathering the DNA samples and getting the diagnostics that we needed to confirm what we're doing.
Right. So it's one, two. So it's about 2,200
Is what
What
we
I've
owe you
spent.
$2,200
Yeah.
so far.
basically.
Okay, and the club itself has spent slightly over $6,000 on diagnostic testing, necropsies, blood samples, and the collection of those samples.
I just have a quick question for Dr. Swap.
Yeah.
Are you including in that the tests we did in the euthanasia in person, or just the samples sent off? Because there was also in-person stuff.
Right.
Make sure you included that
Oh,
as
I did
well.
not include that.
There's those hospital visit and
Right. All of that.
tests and all
Um,
so
that
make
was
sure you include that in the whole.
okay. That probably is about
itemized invoice.
Okay, so it's 2,900 about.
Okay, so we're pushing $3,000.
Yeah.
So where we are right now, we have funds in the bank that have been collected and been donated to for a long time, but we're already hitting $9,000. And again, from what we're hearing, we likely will spend another 12 times. What are those? Another 1,200. Those are $100 a pass for the
Yeah, those are
low pass.
yeah.
So again, your generosity would be much appreciated as we work diligently to try to find this answer. Does anybody have any other questions for Dr. Casal? We can't thank you enough for
Well, no, thank you for having me.
At this time, I would like to adjourn the meeting. Thank you so much for coming. We'll be around to answer